Students. So, yes, in our previous class from cell cycle and cell division, what all we have completed with the interface is done, right? Yeah, in interface we have clearly dealt with what? G1, S and G2 phase of interface are done, yes or no? Yeah, and then we have also completed all the 4 phases of karyokinesis and cytokinesis in both plant cell and animal cell from mitosis. Okay. So, the entire process of mitosis is also done, fine. Now, we are going to begin with meiosis. In today's class, we will clearly learn each and every stage of meiosis. Post that, we will be doing some objective and we will also be exercising the numerical points to be discussed from this chapter. Okay. So, yes, first in meiosis, once let's have a quick recall, ma. Yeah. Meiosis is also called as what, beta? Yes, very good. It is also called as reductional division. Meiosis is also called as reductional division. So, reductional division in meiosis takes place in 2 phases, right? What is that? Meiosis I and meiosis II. Yes or no, ma? Meiosis I and meiosis II. Fine. Meiosis I, if you look into, we have what? Both karyokinesis I and cytokinesis I. Karyokinesis I and cytokinesis I. Karyokinesis I, we have what, ma? Prophase 1, metaphase 1, parallelly take down, ma? Metaphase 1, anaphase 1 and telophase 1. Okay, prophase 1, metaphase 1, anaphase 1 and telophase 1. In prophase 1, we have 5 substages. What are they, ma'am? What are the 5 substages of prophase 1? Leptotene, zygotene, pachytene, diplotene and diakinesis. Fine. And cytokinesis 1, at the end you will get 2 haploid daughter cells. 2 haploid daughter cells. Fine. 2 haploid daughter cells will be formed at the end of cytokinesis. This daughter cells will next go to what? Meiosis II. In between there is a phase called as what? Interkinesis. Okay. In between there is interkinesis. Post that you have what again? Karyokinesis 2 and cytokinesis 2. Karyokinesis 2, we have what? Prophase, metaphase, anaphase, telophase, which is prophase 2, metaphase 2, anaphase 2 and telophase 2. Then after cytokinesis, at the end, how many daughter cells you have, ma? 4 haploid daughter cells. Clear, right? This is the flow chart that you have to remember with respect to meiosis. Let us exercise each stage step by step. What are the events that are occurring in the each stage? So, meiosis, first we have to look into what? Karyokinesis 1, that is prophase 1. Okay? Karyokinesis 1, prophase 1. In prophase 1, what is the first stage, ma? What is the first stage in prophase 1? Tell me, what is the first stage? Leptotene, right? So, in leptotene, what is the events that are occurring? First is the chromatin gets untangled to what? Chromatids, which are thread like structures. This you will call it as untangle or detangle. Okay? And this chromatids will be adjoined to what? nuclear membrane, and so this stage is called as what? Spireme stage or bucket stage. Spireme stage or bucket stage. Fine. And the second event that is happening in the liptotain is centrioles will start moving to the opposite poles like this. Centriole starts moving to the opposite poles. So, what are the two events of the liptotain? Chromatin gets untangled or detangled to chromatids, and this chromatids adjoin to the nuclear membrane, which is called as spireme stage or bucket stage, and then the centrioles or centrosomes will start moving towards the opposite poles. These are the events of liptotain. And then is zygote. In zygote, what happens is homologous chromosomes will form a synaptonemal complex. First of all, you should know what are homologous chromosomes. Any idea what is homologous chromosome? See, look at it, ma. For example, first in the G1 phase, in meiosis, let us observe, meiosis happens in the germ cells, right? So, here you see the basic framework I will give you. Yeah. So, here you look at it, in the interface we have G1, S and G2 phases, G1, S, G2 and here M phase, okay. In G1 phase, let us assume a haploid set of chromosome will enter for the formation of the zygote, right. Let us assume one haploid set X from female and haploid set X again from male. Okay. Of course, chromosome structure you will clearly see in metaphase, but for your understanding I am telling you. Okay. Again, this chromosomes DNA gets doubled in the S phase, which is like this. Yes or no? Fine. Now, during zygote, what happens is those chromosomes which have same alleles located at C. Oh, this chromosome is what? The compact or condensed form of DNA. Yes or no? Compact or condensed form of DNA is a chromosome. Over the DNA, what are present? Genes are present. Fine. Over the DNA, genes are present. Each gene is having how many alleles? 2 alleles will be there. And each gene will be coding for how many phenotypes? Usually one phenotype, one genotype for one phenotype. Okay. So, as such, let us assume for hair color, these are at the same location. For skin color, these are at the same location. For height, these are at the same location. Okay. For retina length or for eyebrows, these are at the same location. Okay. So, those alleles which are located at the same loci of a single gene, those chromosomes are called as what? Homologous chromosomes. Such chromosomes will come adjoining to each other like this, forming what? Synaptonemal complex. So, synaptonemal complex formation takes place in which stage of prophase 1 ma? Zygote. The moment you listen synaptonemal complex, the moment you listen homologous chromosomes, you have to go for the substage, that is zygote. Okay. So, here it is what? Synaptonemal complex. Synaptonemal complex formation happens, synaptonemal complex formation. Okay. As such, in a single synaptonemal complex, how many chromatids will be there? Beta? 1, 2, 4 chromatids will be there. So, as it is a set of 2 chromosomes, okay, it is called as bivalent. As it is having 4 chromatids, it is also called as tetrad. So, here one bivalent is equal to one tetrad. Yes or no? Yeah. So, here in the liptotain, what you have to remember, ma? Chromatin untangling to chromatids, adjoining to the nuclear membrane, where you will be calling it as bucket stage or spireme stage. And then in zygote, if you see, okay, synaptonemal complex formation happens, okay, in between 2 homologous chromosomes. What are homologous chromosomes? Genes present at the same loci of 2 chromosomes are homologous chromosomes. That happens, see. in this homologous chromosomes, these two are called as what? Sister chromatids. Sister chromatids. These two chromatids are called as what? Non-sister chromatids. Non-sister chromatids. Clear, right? Sister chromatids and non-sister chromatids of homologous chromosomes. So, homologous chromosomes, synaptonemal complex, bivalent, tetrad, you will be observing in the zygote. Clear right now, beta? Now you see, in packetine what happens is, in packetine, these two chromosomes with alleles at the same location, right? They will form what? Two non-sister chromatids of homologous chromosomes will start pairing like this. This is called as what? Crossing over. Okay, this is called as crossing over. Crossing over or recombination is one and the same. Here you have to remember one point in connection with your genetics, ma'am. Here I have marked a few alleles. Okay. The recombination between this first gene and last gene is easy than recombination between these two. That is, more the length or more the distance between the genes, more recombination is possible. Less the distance, less recombination is possible. Understood? Yes or no? More the distance between the genes present on the chromosomes, more will be the recombination. For example, here you see, let us assume here we have again 4 genes. Okay, crossing over between these two is easy, but crossing over between these two not possible because the distance between the genes is less. So, here you can also take down this point, more the distance between the genes on a chromosome, more the distance between non-sister non-sister chromosomes. Crossing over always happens between non-sister chromatids. Yes, non-sister chromatids of homologous chromosomes. See, this is one homologous chromosome, this is one homologous chromosome. Crossing over is happening between two non-sister chromatids of homologous chromosome. That crossing over is also distance mediated. More the distance between the genes, more is the possibility of recombination. Less the distance, less will be the recombination. Till here it is clear, right? Next is what? Diplotin. Diplotin you observe ki the very first phase of diplotin is dissolution of synaptonemal complex. The synaptonemal complex whichever is formed, that is both are adjoined like this. Now after chromosomal pairing between non-sister chromatids, this synaptonemal complex will be dissoluted, yes or no? So the very first stage of diplotin is what? Dissolution of synaptonemal complex. Dissolution of synaptonemal complex. Fine, dissolution of synaptonemal complex. And the second thing is formation of chiasmata. It is as simple as it is, nothing to complicate. See, synaptonemal complex is like this, na beta. After crossing over, they get like this. This X-shaped structures are called as what? Chiasmata. Okay, and at this stage only mammalian oocytes will be stopping their cell division at this stage for years together. Okay, during the embryonic stage only hundreds of oocytes will be formed, mammalian oocytes will be formed. Once they reach puberty, every month one egg cell will be released. Okay, that cycle you know from human reproduction. Okay, so mammalian oocytes will stop their cell division at which stage? Diplotin. Okay, so that is one example you take down. Mammalian oocytes. Till here it is clear, Nama? Next, Last stage is what is diakinesis. Diakinesis is the last substage of prophase 1. What is happening over here? First, this starts with disintegration of chiasmata or dissolution of chiasmata. That X-shaped structure will be dissoluted. So, this is disintegration of chiasmata. And the second is what is dissolution of or disintegration of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi. As such, the cytoplasm will get dense. Okay, disappearance or disintegration nothing but they will lose their structural stability. Okay, they will lose their structural stability, because of which the cytoplasm will get dense. Till here it is clear, Nama? Any doubt anywhere? Yeah. Next is what is mitosis. Once we will quickly revise all the 5 substages of prophase 1. In the very first stage, what is that? Leptotin. In leptotin, what is happening, ma? Formation of chromatin will get untangled to what? Chromatids, formation of chromatids, spireme stage or bucket stage, and then centromeres will start moving to the opposite poles. That is the first event that you have seen. Okay, and in zygote, formation of homologous chromosomes, synaptonemal complex formation, and this homologous chromosomes coming together, that phenomena is called as what is synapsis, and the complex is called as synaptonemal complex. These 2 chromatids of one chromosome are sister chromatids, and 2 chromatids of 2 homologous chromosomes, 2 different homologous chromosomes are called as non-sister chromatids. Okay. Before this, you should have a clear idea on homologous and non-homologous chromosomes. Okay. And then we have learned 1 bivalent, here it is equal to 1 tetrad. Next, in packetine, crossing over or recombination is in between non-sister chromatids of 2 homologous chromosomes. Okay, and here more the distance between the genes, more will be the recombination. Last is what, ma? Diplotin. Diplotin is nothing but dissolution of synaptonemal complex, first event in the diplotin. Second is formation of chiasmata, and at this stage only many cells will stay or stop their division, for example mammalian oocytes. And then diakinesis, disintegration of chiasmata, and then dissolution of nuclear membrane, nucleolus and endoplasmic reticulum, Golgi bodies, because of which cytoplasm will get thick. Okay. So, this is the end of what? Prophase 1. Next is metaphase 1. Metaphase 1 is similar to the metaphase of mitosis, but the only difference is here you can see 2 metaphasic plates, right? Okay. So, here bimetaphasic plate is formed.
There is only one metaphase.
Yeah, bimetaphasic plate. Bimetaphasic plate is formed, wherein the sister chromatids are present, right? In a homologous chromosome, 2 sister chromatids will be aligned at the center, and you know all the chromosomes aligned at the center, this is called as what? Congregation. And here opposite asters are present. Spindle fibers will connect to what region of the chromosomes? Kinetocores of the chromosomes. And in metaphase, spindle fiber length is long. Okay, spindle fiber length is long.
In both metaphase.
In only metaphase. In anaphase, spindle fiber length will be reduced. Here spindle fibers length is long. Fine. Next, anaphase 1. In anaphase 1, you see there is no centromere splitting in the anaphase 1. The 2 sister chromatids will start moving towards the opposite poles without any centromere splitting, and here if you see, the spindle fibers length will be decreased. Okay. So, anaphase 1, what you have to remember, ma? Anaphase 1, no centromere splitting. No centromere splitting. Spindle fiber length decreases. Clear, right? And next, here you see telophase 1. Telophase 1, wherein reappearance of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies, and as such 2 daughter cells will be formed, okay, and chromatids get tangled again to chromatin, okay. So here again chromatids gets tangled to chromatin and reappearance of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies. Clear? With meiosis 1. After meiosis 1 there is a phase called as what? Interkinesis. In interkinesis, whichever RNA or proteins are required for meiosis 2 to happen will be synthesized in interkinesis stage. Cells are metabolically active but DNA will not get duplicated. Take down those points. During interkinesis, RNA proteins required for meiosis 2 are synthesized. Fine. And second is cells are metabolically active.
In all stages, cells are metabolically active only.
Yes, metabolically active only. But here we need to specify that cells are metabolically active but then DNA duplication is not happening, okay, or any other cell organelle duplication is not happening. We need to specify that, okay. So cells are metabolically active, no DNA duplication, because this is a gap phase na beta, in between meiosis 1 and meiosis 2 we need to specify all the events. No DNA duplication or cell organelle duplication. Fine, clear till here? Next, in meiosis 2. Meiosis 2 we can also say, meiosis 2 we can also term it as equational division, because it will be same as that of mitosis. Same as that of mitosis. Meiosis 2 is same as that of mitosis. Fine. Here we have prophase 2, metaphase 2, anaphase 2, telophase 2 and then 4 haploid daughter cells will be formed. Look at here carefully. First is prophase 2, the same events whichever are happening in mitosis will happen. What are those? Chromatids formation, spireme stage and then disappearance of nuclear membrane, nucleolus, endoplasmic reticulum, that and all will happen in the prophase 2. Metaphase 2, there is again single metaphasic plate formed at the equator, single metaphasic plate. In the anaphase 2, same like that of mitosis, centromere splitting occurs. Centromere splitting occurs in the mitosis, in the anaphase 2, centromere splitting occurs. Telophase 2, 4 daughter cells will be formed. Same, what are the events of telophase 2? Reappearance of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies, and after that chromatin into chromatids. Okay. Lastly, 4 haploid daughter cells. 4 haploid daughter cells will be formed. These are the complete events of meiosis 1 and meiosis 2 beta. Now you see the differences between mitosis and meiosis, then I will give you a numerical approach of mitosis and meiosis. Look at the differences carefully. Mitosis will occur in what type of cells, ma'am? Somatic cells. Okay. Meiosis occurs in germ cells. And then we have nucleus divides only once over here. Here 2 karyokinesis will happen, right? 2 daughter cells are formed over here without any change in ploidy, but here 4 haploid daughter cells are formed. Daughter cell's ploidy is haploid, or you can also say the ploidy is same as that of parent cell. Here the ploidy is haploid. This occurs, mitosis occurs more frequently in the somatic cells. Meiosis occurs less frequently because it occurs only in the germ cells. Okay, and daughter cells form what? Somatic organs, and here daughter cells form what? Gametes. There is only one prophase, metaphase, anaphase and telophase. Here you have two sets, and there are 5 substages in the prophase 1. And after this, number of chromosomes are not changed in the daughter cells. Number of chromosomes are reduced to half. No crossing over in chromosomes here, but here crossing over occurs in which substage of prophase 1? Good, packetine. And then this is called as equational division, this is called as reductional division. Clear? Any doubt anywhere, ma? Next. Here you see how many chromosomes, before this I will tell you, wait, something to discuss more, just a minute. Mitosis. Look at here now. How many number of divisions occur to form n number of cells, if that is the question? How many number of mitotic divisions to form n number of cells? Now, n minus 1. Okay, where n is the number of cells. Okay, n is what? Number of cells. Let us take for example. For example, 64 cells formed, 64 daughter cells are there. How many mitotic divisions occur to form 64 cells? 64 minus 1 will be the answer, that is 63 cells. And next is this test tube type of question, test tube or cup type of question, wherein in a single test tube 1/4 of bacteria is present. Okay, 1/4 of bacteria. How much? 1/4. In this each bacterial cell division time is, let's take 2 minutes. Okay, and to fill 1/4 it took 16 minutes of time. And in this each bacteria is again taking 2 minutes of time for division. Fine. To fill the complete cup or complete test tube, how much time the bacteria will take? More. 4 minutes. 4 more minutes. Very good. So, on total it is in 20 minutes it will completely fill the test tube. Nothing like, see, 1/4 is already filled. Each bacterial division time is 2 minutes. So, in the next 2 minutes, 16 plus 2 minutes, it will fill half of the test tube. Again 2 more minutes, it will finish the complete test tube is filled. Next major important question is on the generations, number of generations. Okay, number of generations. Fine, number of generations, that is one question. Wherein here you see carefully, how many number of generations does a bacteria take to finish this many divisions, if that is the question. The formula applied is 2 to the power of n, where n is the number of generations. Okay, this is one generation problem you have to exercise. And next, from the meiosis, number of meiotic divisions occur to form these many number of cells. Number of meiotic divisions. Formula is n by 4. Okay, where n is the number of daughter cells. For example, 64 cells are there. How many meiotic divisions happen to form 64 cells? 64 by 4. What will be the answer? Tell Ray. In point we should not sell. Some 14, right answer? It will be some 12 point, like you can take 13 cells or 13 divisions. Fine? 16, exactly. 16 divisions. 16 meiotic divisions will happen like that, okay? And then completely brains are off today, I don't know why. Saturated with exam or what? Next is how many number of fruits, seeds, grains are formed. Like how many meiotic divisions occur to form these many number of seeds, fruits and grains. For that, number of divisions formula is n plus n by 4. Fine, n plus n by 4 will be the formula. So, these are the formula that you have to remember with respect to mitosis and meiosis. Clear, right? Any doubt anywhere? Yeah. Next, here you see how many chromosomes will the cell have at G1, after S, after M phase respectively, if it is having 14 chromosomes at the interface. Same, right? Yes, because how many chromosome number will not double, chromosome number will be the same. It is only DNA that is getting doubled in the S phase. So, here our answer is option B. Next, here you see ma, how many meiotic and mitotic divisions need to occur for the development of mature female gametophyte from megaspore mother cell in an angiosperm. From megaspore mother cell he is asking. 1 megaspore mother cell, this is the question which is asked in 2025. All the time I also stretched you out when I am explaining, okay. Cell cycle, cell division questions will be connected with sexual reproduction of flowering plants or with the ploidy of plant kingdom, and they will ask you the questions. This is a connecting dot between cell cycle and sexual reproduction of flowering plants. So, here inside an ovule, first 1 megaspore mother cell will be formed, right. This megaspore mother cell undergoes reductional division forming 4 haploid megaspores, of which 3 will be degenerated and 1 cell undergoes 3 sequential mitotic divisions, which forms an embryo sac with 7 celled 8 nucleated condition, okay. Here at the micropylar end, synergids and egg cell, which is an egg apparatus. At the center, it is polar nuclei and antipodals at chelicell end. Fine. So, 7 celled 8 nucleated condition. How many? So, this is the female gametophyte, right? To form a mature female gametophyte from megaspore mother cell, 1 meiotic division, 3 sequential mitotic divisions occur, 1 meiosis and 3 mitosis. Option 1 will be the right answer. Next question. Statement based appeared in the year NEET 2024. Look at it and answer. Given below are 2 statements. Statement 1. Chromosomes become gradually visible under the light microscope during leptotene stage. Yes or no? Correct. Next, statement 2. The beginning of diplotene stage is recognized by dissolution of synaptonemal complex. Yes, diplotene is by synaptonemal complex dissolution. So, both the statements 1 and 2 are true. Direct NCERT liners. Next, look at it. Arrange the statements in the correct sequence. Thin thread-like chromosomes with beaded appearance of recombination nodules in pachytene. First you note down the stages. Formation of bivalent in zygotene, good. Terminalization of chiasmata in diplotene. Appearance of chiasmata in terminalization of chiasmata in diakinesis. Appearance of chiasmata in diplotene. Thin thread-like chromosomes with beaded appearance in leptotene. So, here you see 1, 3 and 2 and then 4. See 1, 3, 2,
And last.
yeah, good. Option D will be the right answer. Next. Last question of today's session.
Which of the following correctly drawing figure UPS of this cell? Number of pair homologous
How many homologous pairs are there? 3. Very good. 3 homologous pairs. How many chromatids are there?
6.
Think and say. Chromatids he is asking.
Chromatids 12.
12 chromatids. How many centromeres are there? 6 centromeres.
3, 12, 6. B is answer.
Yes. Option B is the right answer. So with this we have dealt completely with what? Cell cycle and cell division. Okay, in our next class we will begin with biomolecules. Fine, right ma? Any doubts anywhere you can approach me, and online students you can drop a message in comment section, I will be answering. Yeah, thank you.